Well, it's my great pleasure to introduce my friend and colleague, Gonzalo Guirabet, who is the Alexander Agassiz Professor and Curator of Invertebrate Zoology here at the Museum of Comparative Zoology. By now, Gonzalo is familiar to many of you who frequent these talks. This is actually his third Natural History Museum talk, and so I will keep my introductory remarks short. Now, I could give sort of the standard introduction going over his academic and life details, born in Spain, undergraduate and graduate degrees in Barcelona, a postdoc at the American Museum of Natural History in New York before arriving here at Harvard in the year 2000. I could list the extraordinarily long list of papers he has published, many of them very influential and many in the very top journals in the field. I could mention the incredibly long list of big money grants he's received, including one very recently, or the long list of honors, most recently a Guggenheim Fellowship to finish a book. But that does not really capture who Gonzalo is or what he does. To do that, I think you need to appreciate that even as he has been at the very forefront of using molecular and now genomic techniques to understand invertebrate evolution, even as he has been pioneering these approaches in the lab, he has at the same time had an extraordinary program of research in the field. In just the last 5 years alone, he has conducted field studies in Australia, New Zealand, Brazil, Spain, Portugal, South Africa, Mexico, Panama, Belize, the Amazon, and most recently, Antarctica. And I suspect that we'll hear today that I've managed to miss a few of them along the way. And what is it that he's doing on these expeditions? What is he studying? Well, he's studying basically everything, or at least everything without a backbone, from spiders to mollusks to worms and everything in between. Gonzalo studies them all. In fact, I think it's no exaggeration to say that at this point, Gonzalo is the world's leading authority on invertebrate biodiversity. Now finally, on top of all of this, Gonzalo is a world-class windsurfer, regularly competing in tournaments in far-flung destinations around the world. Now, as you know, our series this spring has focused on evolution on islands. If there's any theme that runs through Gonzalo's research, it is the importance of the Earth's deep time geological history in explaining patterns of biological diversity. Tonight, Gonzalo will show how this perspective of continental drift and Earth history enlightens our understanding of island evolution. So please join me in welcoming Gonzalo Guirabet, who will speak on From Supercontinents to Islands, Evolution in Motion.
Wow, that's quite the introduction by the master of islands, and it's going to be hard now to stay on top of my talk. But it's a great pleasure to be here again, talking about some of the research that not only me, but a lot of the people in my lab do. I mean, a lot of the results, a lot of the research that I'm able to do, it's because we have a big team, a great team of people, all of whom work, as Jonathan said, from genomics to going in the field and collecting the very animals that we need for our research. I think that one of the things that really characterizes our research program, or the program in my lab, is that we really need to go and identify and describe the biodiversity that we use for the research that we do. Today I'm going to focus on one aspect of the research in the lab, which is related to islands. Now, you've heard a lot of things about islands in this series, and many of those things, like the odd animals that live in islands, these flightless giant birds and insects that don't fly anymore. We'll see the ground foraging bats that behave like a mouse, for example, or sunflower plants that become trees instead of being like small woody plants, plants there. We'll see these hogs that Darwin referred to in his books, that they were so tame that he could knock them down off the perch with the butt of his gun. Or we'll see things like the giant shrews that lived in the Mediterranean, or the dwarf elephants that lived in many places, in many small islands, from the islands off the coast of California to a lot of the Mediterranean islands. So you've seen some of these themes, and these themes are relevant to all types of islands, no matter what origin they have. Another theme that we see in islands is the theme of extinction. And this has to do, for example, New Zealand, one of the islands or the island systems we're going to talk about today, that it has very unique faunas. And from those faunas that it has, the only mammals that really lived in New Zealand are bats. But those bats became adapted to have a very different behavior from the bats in other places because there were no, for example, mice or things like that. So many of them are foraging, they're just in the ground. But of course, as soon as men got there, so many of these species, or at least the largest one of these species, became extinct. The picture on the lower left that you can see there is the last photo of one of the members of these species that went extinct in 1967. The theme of extinction is very common in islands, and it has to do with the structure of the biology of the organisms that live there, that they adapt to very specific conditions, many times without predators, and as soon as there's an invasion, man-mediated or not, things just change very rapidly. You've seen probably in many of the talks, the early talks, themes related to oceanic islands. Those islands that emerged de novo from the middle of the ocean, whether it's from a plume in a volcano or whether they're formed in the coral reef areas. And these islands have a series of conditions that make their biota to be what it is. For example, when they emerge de novo from the ocean, they're bare. They don't have any organisms living there. So one of the first things that we see in these types of islands is that the animals and plants that make it bare, they're organisms that have the capacity to disperse to these islands. So dispersal plays a very important role in shaping up the biodiversity of those islands. And that's why it's not uncommon to find things that disperse very well. And then once they get to those islands, they might radiate and give origin to some of the biodiversity that we find there. So dispersal plays a key role in short ecological times. But then once the things arrive there, since there's a lot of empty niches, many of the groups that are not very diverse in the centers of origin, they might diversify in these islands and give rise to these evolutionary radiations that are so famous in many of those islands. So one of the patterns that we find in these islands is that some of the organisms that can fly, for example, birds and a lot of seeds that disperse very well through water, make it to all those islands. And then the things that don't disperse very well, they might be able to make it to closer islands. And you have things in between, and some of these graphs show you that some animals that don't disperse very well, if they attach to birds or seeds or some other forms of vegetation that can actually float, they will also make it to far distant islands. Okay, so depending on the properties of those organisms, they're going to make it to those islands or not. So the first things that arrive to many oceanic islands are going to be birds, are going to be flying insects, are going to be ballooning spiders and things like that. And this has been tested in many groups of organisms. This is a spider of the genus Tetrachnatha. And a researcher who gave one of the lectures here, Rosie Gillespie, has shown that as you go farther away from the source of origin of these things, the more distant islands start having more endemicity, more species that only live in those islands. So Hawaii is one of the most remote archipelagos in the world, and pretty much all the spiders of the genus Tetrachnatha that live in Hawaii are endemic. They only occur in those islands. And if those are closer to other sources, then you might have a lower level of endemism. So the common pattern that we see there, for example, also in Hawaii, is that one ancestor of the radiation of honey creepers made it there, and then once they get to the islands, they start diversifying, the habitats become heterogeneous, they adapt to specific foods, and you end up having these beautiful radiations that we've heard about all the time. And for example, also another radiation is what Jonathan Lossos has been working on in the Caribbean and all this, how they've diversified those islands, how you have the same ecotypes that evolve in different islands, and once these animals make it into those bare habitats. The types of islands that I'm going to talk about today are quite different. We call them continental islands or fragment islands. These are islands that originate by one big landmass fragmenting and giving origin to a smaller landmass. We've talked about New Zealand, for example. We'll see some other ones. And these islands have very different characteristics. When they form, they're not bare, right? They're carrying a lot of the biodiversity that was originally in the continent from when they fragmented. And they undergo several processes, some of which are related to the other islands. For example, there's a process called relaxation that not all the lineages that made it into that original fragment are going to make it through evolutionary history. Some of them go extinct, and others, because they have then more availability of habitat, might also radiate. So we might have other types of radiations, for example. One of the phenomena that we're going to talk about is the formation of what is called paleoendemics. Okay, there are endemic organisms that live in those islands. Those are relics often, or we refer to them as relics. They're all lineages that were inherited by those islands. Sometimes they disappear everywhere else in the world, and they make it a single species or very poor diverse groups that only survive in those islands. And we'll talk also about some of the other, since these islands are much older in origin in general than the islands that formed de novo in the middle of the ocean, we might have lineages that are also very old in time. So what are some of the major continental islands? Well, if we go to one part of the world, if we center here in Southeast Asia, we see a lot of islands. And in fact, in this map, we can see most of the major continental islands in the world. We have Madagascar here, off the coast of Africa. We have Sri Lanka here, south of India. We have many of the islands of Southeast Asia, like Sumatra, Java, Borneo, Sulawesi. But not all the islands in Southeast Asia are of continental origin. For example, most of the Philippines are islands that are formed de novo, okay? Most from volcanic origin, some of them from core origin. There's some Philippine islands like Palawan that is actually continental, and it was once connected to Borneo. And then we have Papua New Guinea, which fragmented off Australia, and some of the islands that we've done most work in my lab, like New Caledonia, which is here, and New Zealand. Poor New Zealand is always off the map. It's always really hard to look at. So just to summarize the difference between these types of islands, what we call island fragments or the continental islands, they originate by fragmentation of a landmass that it existed. Okay? And this fragmentation, as the island separates, takes the biodiversity that live there, goes through some ecological processes like relaxation and formation of new species, and continues its evolution. Okay? The difference with the oceanic islands is they emerge from nothing. Once you have those islands, they're empty, and they start receiving organisms by immigration, and then the processes. The first arrivers normally are the ones who might be able to make it and to diversify. Not always, but that's the case. So I want to talk about a few processes that are related to some of these continental islands today. One is continental breakup. This is a process that is very important to understand the biodiversity of these islands. The other things that we're going to talk about a little bit is paleoendemics and this concept of relictualization, relic taxa that have survived in some of these places, and perhaps a little bit about higher level endemism, things that are, like in oceanic islands, normally since they're relatively new, you might find that suddenly the honey creepers have many species, or the anoles have many species. But those are still the same genera that you find in the areas where they originated. While in these oceanic islands, since they're very old in general, what you have is not new species within a group, but you might have genera or families or orders. So this is what I refer to with very old lineages that have persisted there. This is one view of the world nowadays. And the world is formed by these tectonic plates. And those tectonic plates are very dynamic. This thing is moving all the time. Some of them are very stable. It's very unlikely that you're going to have an earthquake in the middle of the Eurasian plate. But it's very likely that if you're along this area where these two plates are creating friction, you're going to have lots of volcanoes, lots of earthquakes, tsunamis, and these things. Okay? And this is very well known. In fact, that area of the Pacific is called the Ring of Fire because there's a lot of seismic activity going on there. And this is telling you something about the dynamic nature of the planet. The continents are very old. So we have continents that have rocks that are 4.4 billion years old. Okay, so some of these areas have been maintained for a long time. And the collision of these plates create things like, for example, when the Indian plate collided against the Eurasian plate, it went very fast, collided with a lot of energy, and formed the highest mountain range that we know nowadays, the Himalayas, for example. When we look at the ocean floor, the story is very different. The ocean is very young. So what this graph is telling you is, for example, here, there is an area where you have new seafloor being formed in the middle of the Atlantic. Okay? And this is actually where you will find a lot of the oceanic islands that emerged in Novo. And the same thing if you go to the Pacific plate. Red means very young. And as you get away from the area where the seafloor is forming, the ocean is getting older and older. But you see that the edges of the Atlantic, when you look at this scale, they're in the order of 100 million years. I know 100 million years is a lot of time, but compared to the 4.4 billion that we have in some terrestrial rocks, it's actually very young. And in fact, there's very little seafloor that is older than 100 million years. And there's a little bit here in the Tethys that gets down to 260 to 180 million years, but most of the seafloor is very young, which means that what's happening all the time is that as these continents reconfigure, there's new seafloor that is being formed, and when it collides with the continent, since it's denser, it goes underneath and it gets absorbed by the crust. Okay? And that cycle goes on all the time. What that cycle is doing is that the continents don't look like they do nowadays through their entire history. So if we go back in time to this area around the the Paleozoic, so you have that most of the southern continents form this large landmass that is called Gondwana. Okay? And as you go about 120 million years, Gondwana started splitting, first with the opening of the Atlantic Ocean, and then it kept separating. Here you have about 99 million years ago until 67 million years ago, when the continents are beginning to look a little bit more like we know them nowadays. So what I'm trying to do is study the biota, mostly the invertebrates that live in these land masses, and see how they've been reconfiguring as these continents move apart. And we're especially studying a lot of the taxa that live, a lot of the invertebrates that live in some of the island fragments that were left after the breakup of Gondwana. Yeah. And the breakup was very important in many ways. We can see here, for example, the Indian subcontinent breaks up from the north part of Antarctica and starts going north very quickly. It leaves Madagascar behind and then keeps going north until it collides with Southeast Asia, for example. Okay? And if we look from another perspective, one that has interested me a lot is the connection of the southern continents. We see here the world look from Antarctica. When we go about 50 million years ago, there was still a connection between South America, Antarctica, and Australia. Antarctica was in the South Pole, but it wasn't as cold as it is today. So there were a lot of animals and plants that lived over there. In fact, Antarctica is that cold, not because it's in the South Pole, which in part it is, but it's because after a certain time, these connections broke, this one and this one here. The Drake Passage opened up and created the circumantarctic current that is being isolating thermically Antarctica from the rest of the oceans. There's very little water mixing, and it's been creating basically a freezer. It's isolating it from the rest of the world. And that's when Antarctica became completely frozen, and a lot of the biodiversity from there disappeared. So all the animals, many of the animals that I study, they live in what I call circumantarctic Gondwana. So the land masses that were once touching Gondwana, this is South America, this is South Africa, Madagascar, India has gone north, Australia, New Zealand. And these things were connected to New Zealand, to Antarctica, and many of the organisms that we study nowadays did live in Antarctica. We just don't find them alive there anymore, and we cannot find the fossils because the fossils are under sometimes kilometers of ice. So it's very difficult to get to the rocks in Antarctica to look for some of these things. And one of the things that we try to do when we find a distribution of organisms that live around Antarctica, we tend to say, well, these organisms are found in these temperate Gondwana, Therefore, they originated there, and they've been, breaking up with the continents, and they haven't moved around. And we used to explain that these distributions are telling us something about the history of these groups. Now, this is not technically correct. Okay? We can draw a cladogram, the relationships of these organisms that live in these land masses, and we can use techniques like looking at morphology or looking at molecules and generate these schemes, these things, we call them phylogenetic trees, and they're telling us that, for example, the animal A and B have a common ancestor. They were related. Okay? And we can say, well, A lives in Chile or South America, B lives in South Africa, and then we can translate that taxon cladogram into what we call the area cladogram. And it's telling us that for that organism, there's been a connection between South Africa and South America, for example. And normally, when we see those distributions, we say, this is because these things had a Gondwanan distribution originally. They've been breaking apart with the continents, and that's what we find those. But nowadays, that's not necessarily the case, because we could also explain the same distribution if these organisms had originated here and they had been circulated around with the circumantarctic current, and they've been distributed along all that. So the fact that we find organisms living in the former Gondwanan continents doesn't mean that they have evolved there. So what we're doing nowadays to study biogeography is not only looking at the distributions and the cladograms of these organisms, but we need to apply some type of fancy techniques to try to come up with the age when those distributions were achieved. And once we have the age, we can say, look, this distribution was acquired before the continents broke. Therefore, that distribution is the legacy of the continental breakup, for example. As opposed to saying, look, this is too young. The diversification of these things has occurred in the last 10 million years when the continents were already shaped like that. Therefore, we cannot say that that is a distribution that was achieved by continental breakup. And these are the models that we do. For example, we have a landmass that was once continuous, and we're going to call the red part of the landmass and the blue part of the landmass. And that landmass breaks up into two, and it separates. Okay, now there's C in between, and those two landmasses keep separating. And we might find three types of distributions that are actually congruent with those landmasses. But depending on the timing of those distributions, the evolutionary history of those groups is completely different. For example, here, the group originates when the two landmasses separate. Okay? And then they diversify each one on one of those landmasses, and we call that thing vicariance biogeography, for example. We might have examples where the origin of the groups is prior to the breakup. The group starts forming, and then they get carried over by each one of those landmasses and continue diversifying. And for us, it's very difficult to distinguish these two scenarios unless there's a very long history before the breakup. But we might find another distribution which is perfectly the same or equivalent to that one, except that the group has originated after the two landmasses separate. And therefore, we cannot say that that distribution, which matches that Gondwanan biogeographic scenario, is actually the consequence of the continental breakup. The only explanation that we can give here is that the group originated after the continents broke, and the current distribution has been achieved a posteriori by dispersal, for example. Okay, so that's what we're trying to do, and I'll bring that back to our research in a little bit. We were talking about some other things, often islands, we're talking about paleoendemics and relict taxa. And some of those are very prominent in a lot of the discussions on biogeography. This is Amborella. Amborella is a very weird flower. It's a flower that lives only in New Caledonia. And New Caledonia is this tiny island which is part of a larger continent called Zealandia. Why is Amborella relevant at all? A little flower, not really pretty, or not very pretty. Well, it's very interesting because there's one species of Amborella, and it's the sister group to all other flowering plants. This means that the first flowering plant, when it diversified, gave origin to one lineage, which is Amborella nowadays, or survived only by Amborella, and another lineage that gave origin to all the other flowering plants that you know. Okay? So from the perspective of an evolutionary biologist, this is a very important flower because it's the closest group that you have to the earliest divergence of the flower. So islands often retain some of these groups. Now, can we say, and people sometimes have said, oh, flowering plants originated in New Caledonia because the most basal flowering plant or the first offshoot of the flowering plants lives in New Caledonia. Well, unfortunately, we cannot say that, and I'm going to show you that with another example in a second. So Amborella is a paleoendemic, a relict, a very depauperate old lineage with primitive morphology for flowering plants. And we find paleoendemics that are common in many islands. For example, when we go to New Zealand, one of my favorite islands, we have the moas, which are these giant rat-eyed, flyless birds, and the kiwi birds. We'll talk about the tuatara today, this weird reptile that looks like a lizard, but it's not a lizard. And some frogs that are very old also, and I'll talk later on about some of my velvet worms and harvestmen. Here is the tuatara. I assume many of you know a lot about natural history, and you've heard about this little thing, which we cannot call a lizard. Okay? Tuataras are very interesting. Again, there's one species, and they're like Amborella. They're the sister group to all squamates. Here you have your geckos, your lizards, your snakes, okay, all those things. And then there's this other lineage from the early divergence of these things, which are the tuataras, which today, again, like Amborella only lives in New Caledonia, the tuatara only lives in New Zealand. And we could say the same thing. Did squamates or the ancestor of squamates originated in New Zealand? No, we know that's not true. And we know that because, unlike Amborella, we have a good fossil record of a clade of reptiles that is called rhynchocephalans. And rhynchocephalans were distributed all over the world with lots of fossils in North America and Europe. This is a bias not because they lived here, probably more, but because there's many more paleontologists that have worked in those places. But they had a worldwide distribution. And what's happened with rhynchocephalians is that they have gone extinct everywhere. And because New Zealand got isolated at one point and didn't have many predators, and for the chances of evolution that we know that they don't play by the rules, they just, went from a global distribution to just one species living nowadays in New Zealand. And we don't know for how long it's going to live there because it's been decimated from the mainland. It's now only living in some sanctuaries and in a few islands offshore that are deprived of rats. So this paleoendemic is a very old lineage with very low diversity that has gone extinct in the rest of the world and has survived in an island. So that's why islands, these continental islands, tend to be very interesting because they might maintain some of these paleolineages. But they're not informing us a lot about the origin of the island, about how those groups diversify, because we only have one. We can tell that it's separated from the rest of the squamates about 280 million years ago. But we don't know anything that has been going on during these 280 million years of evolution. We cannot do any molecular estimates because there's no more diversity there. So there's very little information that we get from Amborella or from the tuatara to understand the history, the biological history of those islands. So they're very charismatic. We always talk about them when we work on these islands, but there's very little thing that we can do. And this is why I work on other types of animals. I work on things first that you can collect many, that you can bring them to the lab, and you can sequence their DNA, that they are diverse in those islands. The groups that have diversified, and not only in one of those islands, but in many of them. And they're found in Old Gondwana and land masses, for example. And some of these places have had very troubled history. It's difficult sometimes to understand some of these maps. Here's East Antarctica and Australia, and you have a lot of stuff going on here. And this stuff, it's microplates. And some of those microplates have something here, which will give rise to an island like New Caledonia. And some other parts here that are actually 3 main islands that at one point will merge and form New Zealand. Okay, so we have these organisms that lived in all these land masses that were connected to larger land masses, and that through history they have changed. For example, the history of New Zealand is very interesting for me because you can actually test many hypotheses of island evolution, from very old ones to very recent ones. New Zealand has had a very troubled history that can be summarized by about 80 million years ago. It separated from Australia, okay, by the opening of the Tasman Sea, and then, kept drifting apart. And during about 25 million years ago, went through an episode. It was a global episode that affected New Zealand like many other places, which is called the Oligocene drowning. Most of New Zealand went below seawater. There's a controversy whether the whole New Zealand went under seawater. And that's one of the things that I've been studying through the years, trying to understand whether New Zealand really went underwater at 25 million years ago or not. But whether it went all the way down or not, this is the surface of New Zealand. This will be modern New Zealand, whatever area unit we use. But you can see that during the Oligocene, there was only a little fraction of the total surface of New Zealand above seawater. Okay. In addition to that, about 7 million years ago, started with the orogeny of the Southern Alps, the formation of the Southern Alps that are beautiful, one of the most beautiful mountain ranges in the world. And it formed along this west coast of New Zealand, isolating the east and the west part and giving them very different climate. The west part is really wet, and the east part has become much drier. Also, about 2 million years ago, there was the glaciations that were also global, that affected New Zealand to a big degree. And then even only 16,000 years ago, we're talking now about human times, the North Island and the South Island of New Zealand separated by a very drastic fault movement. Okay? New Zealand is in between two plates, and there was one slippage of those plates, and only 16,000 years ago, they separated those two islands. This is a schematic of New Zealand through all these 80 million years, but they never really looked like that until very recently. This is actually what New Zealand looked like, these very different plates. And probably each one of them originated from a different part of Australia and carrying their own lineages. And we still see some of the history of those lineages in some of the groups that we use, that they have very little dispersal ability. So, New Zealand has changed in shape, even is changing vegetation coverage. This is during the Pleistocene, about 1.8 million years ago. New Zealand was a very large forest and then underwent a much drier period. So you have there many different levels of hypotheses that we can test now with these methods that allow us to give time estimates to the biological events that have happened through the history of the groups. So that's what we're doing nowadays. And one of them that we're testing specifically is this idea. We've seen that New Zealand, about 25 million years ago, lost a lot of its surface going underwater. But a few years ago, about 10 years ago or 9 years ago, there was a new wave of biologists from New Zealand. We're still debating whether they did that to get more grant money or not, but they just said that New Zealand was completely submerged. And that all the biota that we find nowadays in New Zealand, including the tuatara, that has been extinct everywhere else, and many other things, they just arrived to New Zealand in the last 20 to 25 million years, which seems very short time for some of the groups that we have nowadays. At the time, with my former student, graduate student Sarah Boyer, who's now a professor at Macalester College, we wrote a reply saying that, look, we have many candidate organisms that would contradict that hypothesis. But to tell you the truth, at that time, we hadn't done any of these exercises of collecting lots of animals and providing dated phylogenies where we could actually test these things specifically. Because remember, the difference is going to be not on the distributions of the organisms, it's going to be whether those organisms were there before 25 million years ago or were not there at 25 million years ago. Okay, so that's the big difference. They are saying that everything arrived from Australia or from nearby land masses after 25 million years ago. So that's what we want to test. And what organisms do we use to test some of these things? Well, we're looking for very old lineages. So one of the good things of working on invertebrates is that we have animals that are very old. For example, this is the oldest daddy longleg, a harvestman, that actually looks, well, here is flat in a rock, so it doesn't look like much. But what you see here is the ovipositor of the female that has exactly the same structure that we have in modern daddy longlegs that you find running across the yard. And these animals have a lot of the characteristics that are very similar to modern ones. This is a fossil from the Rhynichair that is more than 400 million years old. The daddy longlegs looking quite similar to what we have nowadays. They were, even if it's flat in a rock, already around for 400 million years ago. If you are skeptical with flat rocks, which I understand that some people might be, we can go 300 million years ago. And then we have three-dimensional daddy longlegs. You know, what we have here is a rock. It's a nodule of a rock. And when we scan it with certain machines that we use, the micro-CT scanners, we see what's inside that rock. And there's a differential scanning that picks up different densities in the rock, and then we can reconstruct that with a computer, and we have a perfectly defined three-dimensional animal that we can rotate and look at all the characteristics. We can see the chelicers, we can see the palps, we can see the legs, we can count the tarsomeres on the legs on an animal that you're not seeing physically. It's inside a rock, but it's three-dimensional. And we don't only have one, we have many. We can look at structures like the genitalia of these 300 million year old, and we have many other ones, some of which look very much like some of the daddy longlegs, again, that we find out here running in our backyards. Okay, so this is telling us that these organisms are very old, that they haven't evolved much, and we know for other studies that they've been in the same areas for a long time. We also look at another group of organisms called velvet worms. And this is a paper we have now submitted describing the first unambiguous terrestrial velvet worm. This is from the same sediments that we had the three-dimensional ones. Unfortunately, this is not three-dimensional. But when we look at the structure of these things, the annulation of the cuticle, the little antenna that has here, it looks exactly like the ones from these mother velvet worms. That antenna with these annuli that alternate large and thin ones is very similar to this antenna that we find here. These animals were terrestrial, lived in terrestrial habitats. They are the ancestors of these daddy longlegs. Some of the most beautiful animals that you can encounter if you go around. Velvet worms. And these velvet worms have a very interesting distribution. This is the distribution in green of a family called Peripatidae that is being studied by one of the graduate students in my lab. And then blue, you have this other distribution of the other family called Peripatopsidae, which is very similar to some of the other distributions that I've shown you earlier, these animals that lived in Antarctic Gondwana, or what I call the temperate Gondwana. And so we're going to focus a little bit on those ones. And a couple years ago, we published a study where we're reconstructing the relationships among these organisms and also dating the trees to see if we can test some of these hypotheses about Vicarian's biogeography, about whether New Zealand had diversity surviving this supposed drowning event. Okay? And we're looking at very ancient group, again, more than 300 million years old, and we have collected it along a lot of the distribution places. And we can look at the general tree of the velvet worms, and what we found is that this family, the Circumantarctic Gondwanan family, has a clade, or two clades, two groups, that have a trans-Tasman Sea distribution, meaning that they are in New Zealand and in Tasmania and Australia. Okay? The opening of the Tasman Sea is that event that I told you earlier, about 80 million years ago, when New Zealand separated. And we can follow many of the major splits of each one of these groups, the separation of the two families about 350 million years ago, the diversification of these peripatopsids, which follow very well the splitting of eastern Gondwana from western Gondwana, and then the split of eastern Gondwana into the different land masses. And if we look into New Zealand, more specifically, we find one clade of a genus called Peripatoides that is found both in New Zealand and in Tasmania. And this is at the time, the diversification around 80 million years, which is when the Tasman Sea was beginning to open. And then another group, so it's a repeated experiment of nature, when we have another group of velvet worms also between New Zealand and Tasmania. And these two have actually very different biologies. One of them give birth to little babies that they have in a uterus, and the other one lays eggs. Okay, so there's no way that this could be some sort of like the same lineage we don't have good resolution. These are two old lineages with different morphologies. So what we do with these types of organisms is to study precisely this thing, the continental drift. Actually, we've been working on these things for a long time. A few years ago, Carl Zeimer published an article in the New York Times describing some of our research, and this was about the hitchhikers of the plates. This is something that someone sent me from the Metro News in Dublin, using our photos. This is one of my photos. I don't know where they got it from. They didn't ask for it, but anyhow, it's nice to see your research highlighted in some of the popular press. And they were talking about these animals because this group of daddy longlegs has a distribution that looks very much what we have here. We have a family that lives in the older Laurasian continent, the northern continents. And we have other families that live in the southern continents, one of which is distributed across Africa and tropical South America, and then this other one that lives in circumantarctic Gondwana, South America, Southern Africa, Madagascar, the Indian subcontinent, but only in Sri Lanka, and then Australia and New Zealand. Okay? And these are beautiful examples to do some of these studies. Our big conundrum here is this family, which is endemic to New Caledonia. And it's not related to any of the families that are closest geographically. So this is one of the big things that we haven't been able to disentangle. New Caledonia has a similar controversy to the one in New Zealand, except that it was below seawater until 37 million years ago, and the geological evidence for that is very strong. So we have a lineage that is endemic to New Caledonia, another Amborella, except that this one has many species. Now, we cannot say that those species diversified before 37 million years ago. Our molecular dating puts the diversification around 35 million. With the error bars, we cannot really distinguish that hypothesis. So there is a possibility that this thing had arrived there after New Caledonia re-emerged. When we go back in time looking at some of these distributions, obviously we postulate that they've been acquired through continental drift. So let's go back in time a few million years ago. So if we go to the period of the Cretaceous, where Earth more or less looked like that, you can see that the group that is found in West Africa and tropical South America was, a contiguous group. So it diversified like that. And these groups that were actually in the southern continents around Antarctica, they're getting closer and closer. Those groups had a contiguous distribution before they started diversifying. Okay? Another of my grad students focused on this other problem, which is Southeast Asia, which is really, really complicated. It's one of the most complicated geological systems in the world, with so many islands that have come together and have come apart. And today we're not going to have time to talk about them. But for island biogeography, this is another of the areas that is most interesting. And if we continue going back in time, you can see that these continents at one point became together. And Southeast Asia was not part of Laurasia, the large continent that is most of Asia. Actually, Southeast Asia came from the northern rim of Gondwana, but it just split up very early. So what we find is that these things, when we go through time, end up related to some of those Gondwanan lineages. And with this, we go to all these places. This is what I have to travel as much as Jonathan said, and we need to go to all these places to collect these animals because very few people can collect these tiny little things from the leaf litter, and we need to go there, collect them to have these densely sampled studies when we can represent all the land masses. And this is, for example, the one from New Caledonia that I'm telling you. That is all diversifying very recently, despite having a very long branch. So we could have only one, it will be Amborella. But at least since we have more, we can start testing some of these hypotheses about their diversification. So the one that we've centered most of our attention until now in the lab is the group that lives around Antarctica. And I always say, I will bet anything I have that there were Scyphophthalmi living in the middle of Antarctica. We're just never going to find it. And these are beautiful animals for those of you who like them. For the rest of the people, they're just tiny little brown, indistinct daddy longlegs. Okay? But I love them. I love them because they have all that history written into them. And we've been constructing this phylogenetic analysis with these groups of organisms, and we're able to test once we have a dense sampling. This study is 16 years of sampling these things in all these land masses. And we finally got it published this month, actually. And one of the things that we show here is that from the 3 genera that we find in New Zealand, one is a little bit like Amborella, has 1 species, maybe 2. There's not much we can say. It's an old lineage that relates to some perhaps Australian groups, but there's not much we can say. But for the other 2 lineages that live in New Zealand, one is this genus Aoraki that we named after Mount Cook. Aoraki is the Maori name of Mount Cook. And we can see 2 things in this tree. One is that the group starts diversifying around the Triassic-Jurassic. This is way before the 25 million years of the purported submersion. And also, when we do some analysis of the diversification of this group, we see that the group has been diversifying at a constant rate through time, meaning that it hasn't had weird things in the diversification rates that you will expect by most of the landmass of New Zealand disappearing. When we look at the other genus, which is even more diverse, Rakaia, it originates a little bit earlier. I mean, we have large error bars, but the error bar never even gets close to the 25 million years. So we're talking about groups that have been diversifying at a constant rate for about 150 million years in New Zealand. This is telling me that these groups have ancient cladogenesis in New Zealand, even when it was connected to Australia, and that they've been continued, their evolution in their land masses for a long time. Okay? So we can test this hypothesis, and once we have more and more organisms, that's why we're studying the velvet worms and other groups of daddy longlegs, but then we can say, look, it seems that all these groups are contradicting, are falsifying the hypothesis that New Zealand was completely underwater. Okay? So that's the type of research that we do. Now, continental islands are complicated. For example, New Zealand. We do have groups that have dispersed there. If this is the Oligocene drowning episode, we have molecular signal for many groups telling us that the group has originated in New Zealand at a very recent time and has diversified at a recent time. So the fact that we have groups that have arrived there by dispersal very recently does not contradict that the island was or was not submerged. We have other groups that might have originated before this Oligocene event, but the diversification is more recent. Sort of what I was saying with the other group from New Caledonia. This group doesn't allow us to falsify whether this island was submerged or not, because it could have come from Australia, one arrival here, and then diversify, right? So again, that group doesn't falsify our hypothesis. We have groups that are beginning to get closer, like kiwis and moas. We know they've been diversifying for quite some time, but the diversification did not, again, pass this event. And many of the groups that we're looking for are groups that are much older, that they've been diversifying through this episode of drowning, so we can use those to falsify the idea or the hypothesis that the islands have been completely submerged. Just, I was mentioning earlier that there's systems that are even more complex, where you have not only, New Zealand is still isolated, but in Southeast Asia, you have so many islands, some of continental origin, some of volcanic origin. Some of them, like Sulawesi, is composed of different terrains and different volcanic accretions. So it's really difficult to disentangle what's going on. And then also, the sea level of these things have changed quite a lot in the last few million years. So what looks like many islands nowadays, when the sea level was lower, was a contiguous landmass that it was connected to Southeast Asia. So there was a lot of influence of Southeast Asian taxa. So really working in Southeast Asia is very difficult to be able to find good examples to test some of these hypotheses. But one of my grad students, Ron Klaus, did work there and sampled these things. We included samples from all the continents. And we actually have a very beautiful, similar story of these animals coming from, as I said earlier, from the north part of Antarctica or Gondwana, and then once it docked with Southeast Asia, starts some level of diversification. And we can see that this diversification went in one direction until it found the Himalayas, and then in two other directions, radiating and giving origin to some of this diversity of daddy longbecks over there. Can't really talk about that. I just want to finish with some pretty pictures, because, I think that, and I keep working on groups that were around Antarctica. I've been saying for a long time that I would love to see what's in Antarctica in terms of these groups, and I've said already that I'm not going to see it. But at least what I wanted to do is I was recently in an Antarctic trip to look for other groups and things that can actually help me with some marine organisms to reconstruct some of this biogeography. Now, marine organisms are a little bit more complicated because they tend to have dispersal larvae, but in Antarctica, because of the climate, it has a lot of species that are actually brooders. And species that are brooders tend to have much more geographic structure that perhaps we could use to study some of these connections through time. So I don't have many results, but I have some pretty pictures of some of the places where we were. Just 2 months ago, we were actually diving in these places, collecting some of the animals, doing a lot of intertidal work, which is quite challenging because you're trying to get in between these pieces of iceberg to sample your animals, getting into the water to get our samples, always making sure there's no seals around, no leopard seals around, because they're not. And you know that Antarctica is such a special place. It's the only place where you have fishes where their blood has no hemoglobin, right? Because they can't transport the oxygen. It will freeze the hemoglobin, so they don't have hemoglobin. Amazing landscapes, and there are actually some paleontologists working in some of these places, looking at the origins of, for example, penguins. So there's some vertebrate paleontologists and also dinosaurs. I'm more interested again in some of the invertebrates. These are Pycnogonids. It's a group that is very diverse in Antarctica. We collected lots of animals, lots of these things. Beautiful nudibranchs or sea slugs. I thought trilobites had gone extinct, but there's these isopods in Antarctica that look like trilobites. It's amazing. And all the fauna there is exceptional. This is one ribbon worm, an emertian that I was handling there. They get much larger than this, actually. And with that, I just want to thank everyone from the lab who's helped a lot with all this research, and to you for coming here and listening to this talk. And if you have any questions, I'll be happy to try to answer.